Hi everyone, Dr. Mike here. In this video, we're taking a look at the difference between fed state and fasting state metabolism. Let's take a look. So to begin, we need to understand what's happening in the fed state. This is immediately after ingesting a meal. One important thing that you're going to find in this state is that in your bloodstream, so we're going to focus predominantly on glucose, because it's the primary energy source for a number of important tissues, such as neurons or the brain, such as the retina and the lens, so the eye, also the renal medulla and also red blood cells as well. Glucose is very important. So immediately after a meal, what you're going to find in your bloodstream is your glucose levels are high. That's unsurprising. Now, when I say high, we need a reference point, right? So what is the normal blood glucose range? Normal blood glucose range typically sits between 4 to 6 millimoles per litre. Now, does that really help anyone? At least not within medicine. It's pretty tricky to understand the context of that. All right, let's try and put into context. 4 to 6 millimoles per litre, let's just take the middle of that, 5 millimoles per litre. That is pretty much equivalent to 0.22 teaspoons of glucose per litre of blood. We've got around about 5 litres of blood. So in all of our blood, we've got around about 1 heaped teaspoon of glucose. So hopefully that puts it into context. That's the amount of normal circulating glucose in our entire bloodstream is 1 heaped teaspoon. Now, we've just had a meal, our blood glucose levels go up. So it's going above this 1 teaspoon. It can go to more than 2 teaspoons worth, right? So it could go to 10 millimoles per litre. But that's going too high. We want to drop it back down because we want to maintain homeostasis. Luckily for us, when blood glucose levels are up, it goes to the pancreas, because blood goes everywhere, right? It goes to the pancreas and stimulates the release of a hormone called insulin from a specific area called the pancreatic islet cells, specifically the beta cells there. So that releases insulin. Now, what insulin does in this case is it allows for glucose to leave the bloodstream and enter tissues, specifically or predominantly 2 major tissue types, muscle tissue and adipose or fat tissue. Tissues like the brain and the liver, for example, don't need insulin. They'll simply suck the glucose in from the bloodstream when the concentration is higher in the blood than in the tissue. But because they're not large organs, they can't really drop the blood glucose levels enough. But we need big tissues like muscle and fat, right? That makes up most of our body weight, but they're insulin dependent. And so what they'll do, what this insulin does, is it'll go to the cells and it tells the cell membrane to express a glucose transporter. So here's a glucose transporter termed GLUT, G-L-U-T, and there's different types of GLUT, GLUT 1, 2, 3, 4, so forth. And this then allows for the glucose that's in the bloodstream now to enter those tissues like the muscle and the fat. What happens to this glucose? Well, I want to focus on what happens in a liver cell, because the liver is really important at mobilizing all of these nutrients, storing and mobilizing. So let's now call this a liver cell, a hepatocyte. The glucose has entered, and what will happen is glucose undergoes something called glycolysis. Glycolysis is moving from glucose to glucose 6-phosphate, fructose 6-phosphate, fructose 1,6-bisphosphate, splits in half. Remember, glucose is a 6-carbon molecule, C6H12O6, 6 carbons. And in the process of glycolysis, it rearranges those carbons to make it easier to hand off electrons and protons. I'll talk about that in a second. So as we go through glycolysis, it's 6 carbon to here, then it splits into 2 3-carbon molecules. Now, you'll notice that I'm not spending a lot of time on glycolysis because I've done a video on that. If you want all the information required about glycolysis, watch the glycolysis video by Dr. Mike. And it's 2 3-carbon molecules here. Now, the dihydroxyacetone phosphate, which is sometimes written as DHAP in your textbooks, that will turn into glyceraldehyde 3-phosphate, which means we end up having 2 3 carbon molecules here. So we end up having 2 glyceraldehyde 3-phosphates, which means we have 2 1,3-bisphosphoglycerate, 2 3-phosphoglycerate, 2 2-phosphoglycerate, and then that turns into phosphenylpyruvate and then pyruvate. That's glycolysis. But what's the point of doing this? I said, well, one, it's to remove electrons and protons. True. The other is this. When glucose enters and turns into glucose 6-phosphate, we actually use energy to do this. We take ATP and it turns into ADP. We use energy. Then at this step, we do the same thing. We take ATP and we spit out ADP. So we use energy. Is this what we want in this process? Remember, we're in a fed state. We've got all of this glucose. Then we go down to here, down to here, down to here, and we take ADP and we generate energy. We generate ATP. But remember, we've got 2 molecules here, so we end up taking 2 ADP and generating 2 ATP. We've used 2 ATP here, we've generated 2 ATP here, so we've wiped the slate clean. Then as we go from phosphenylpyruvate to pyruvate, we generate 2 more because there's 2 phosphenylpyruvate, there's 2 pyruvate, so we generate 2 ATP. We take 2 ADP and we generate 2 ATP. Let's just think about this. Let's do the maths. If we have a look at glycolysis and we look at the net energy production and loss, -1, -2, +1, +2, so we've wiped the slate clean, +2. So we've generated +2 ATP in the process of glycolysis. So glycolysis allows for us to generate ATP directly. But in addition to that, it also takes a molecule called NAD+, because there's 2 here, it takes 2 of them, and it generates 2 NADH plus a free-floating hydrogen ion. What the hell does this mean? This is the molecule, NAD+ is the molecule that steals electrons and protons. I'll tell you why in a second. The details of that are in the glycolysis video. So we've also generated 2 NADH. So you can say that the whole reason for bringing glucose into a cell is it generates energy. I'll explain that in one sec. As we go in now to the Krebs cycle, and pyruvate goes to acetyl-CoA, and to citrate and alpha-ketoglutarate, succinate, fumarate, malate, oxaloacetate, what you'll find is this. Firstly, when pyruvate turns to acetyl-CoA, acetyl-CoA must bind with oxaloacetate to form citrate. When citrate turns into alpha-ketoglutarate, it takes NAD+ and spits out NADH plus free hydrogen ions. Alpha-ketoglutarate to succinate will do the same thing. Plus, it generates ATP. Cool. Then we go from succinate to fumarate. That takes FAD, which is very similar to NAD, and produces FADH. And then when we go from malate to oxaloacetate, it generates more. NAD plus 2 NADH plus free hydrogen ions. So what we get here for Krebs cycle, when it comes to looking at the net energy, we've got 1, 2, 3 NADH. We've got 1 ATP, and we've got 1 FADH2. So the Krebs cycle is energy generating. Now let's talk about, we've generated all this NADH, FADH2. This is where the electron transport chain comes into play. What happens here is that let's draw it in a different color. Let's draw it in blue. The NADH and the FADH2 have stolen hydrogen, right? Remember, here's NAD+. It's turned into NADH, so it's stolen hydrogen. Remember, hydrogen on the periodic table is the first element. When it's as a complete atom, it has a positive proton and a negative electron flying around it. The negative electron cancels the positive proton out, so generally a hydrogen atom is just H. But it's got one proton, one electron. Here, NAD+ has stolen a hydrogen. FAD has stolen two hydrogens, because that's supposed to be FADH2 down here. My apologies. It's stolen two hydrogen, which means it's stolen both the proton, which is just hydrogen without the electron, and the electron. So here what it does is it releases the electrons and it releases the protons, the hydrogen without the electron. Now, what this means is these proteins of the electron transport chain embedded in the inner membrane, they take the electrons and play hot potato. As you pass electron from protein to protein, it excites these proteins. It excites them and allows for them to take the hydrogen and pump the hydrogen up into this intermembrane space. So now we've got a hydrogen ion concentration that's high in this intermembrane space. As we know, because of diffusion, these hydrogen want to go down their concentration gradient. They move through this amazing thing called an ATP synthase, which turns this turbine, spin, 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 and that generates a huge amount of ATP. So effectively, the argument you can make, and that I'm making right now, is that NADH and FADH2 make energy. So we can use them as proxies for energy. So at the end of the day, my point here is that in the fed state, what happens is glycolysis and the Krebs cycle make a bunch of energy. It's energy generating. That makes sense. We've just fed, we make energy. But sometimes we don't want to eat and then just use all of those nutrients for energy production, right? We want to use some of those nutrients. So at some point, the energy that we produce here, the high levels of ATP and the high levels of NADH, for example, will say, hey, we've got enough. We've got enough. So it backs up. So if these NADH levels are too high, think about it, it's going to not want to go from NAD plus to NADH because they're already high. It's as high. If the ATP levels are too high, it doesn't want to go from ADP to ATP. Right? So it backs up. And it says, hey, glucose, don't make any more ATP. It tells the glucose to turn into glucose 6-phosphate, which then turns into glucose 1-phosphate. And glucose 1-phosphate can be stored as glycogen. Now, glycogen are basically glucose molecules snapped together, right? So that's a storage form. So we either, in the fed state, use glucose to make ATP, or we store it as glycogen. A lot of this glycogen is stored in the liver, in the muscle, and in the kidney. We store, or at least can store, around about 190 grams of glycogen. And we only need around about 160 grams of glycogen for energy in a day. So we've got around about 30 hours of glycogen available to us. Now, that's not if you're undergoing exercise or intense manual labor. That's just for survival, right? We've got enough glycogen to last us for 30 hours. Less if you're doing a lot of exercise. So that's the fed state. It's mediated by high glucose and high insulin levels. And the outcome of that was high ATP, high NADH, and obviously high FADH2, but also high levels of substrates like acetyl-CoA and citrate. Let's just write citrate. The reason why I'm writing that is because all of these are strong negative regulators, because once they go up, it says we've got enough. So they're strong negative regulators of the Krebs cycle and glycolysis. So that's the fed state. What's now happening when we've fed, but we've stopped eating? It's been 3 to 4 hours post-eating, postprandial. What happens? Well, let's have a think. Our blood glucose levels are going to drop. So let's have a look now. This was in the fed state. Let's now have a look in the fasting state. Again, fasting state is any time after 3 to 4 hours after eating. Our blood glucose levels go down. Cool. Goes down, makes sense. Goes below 4 millimoles per litre. We want to bring it back up. So the fasting state is recognized by having dropping blood glucose levels. When blood glucose levels drop, it doesn't stimulate insulin to be released. It stimulates another hormone that's released from the pancreas called glucagon. And you can remember this because when glucose is low, glucose is gone. Glucagon. Glucagon is released when glucose is gone, or at least glucose is low. It's also going to have low insulin, right? This is important because here, when insulin levels were high, not only did it allow glucose to enter the cell, the insulin allowed for the enzymes of glycolysis to be kicked into action and allowed for the enzymes of the Krebs cycle to be kicked into action. Here, when glucagon levels are high, it inhibits the enzymes at these steps. It inhibits glycolysis. It inhibits the enzymes of the Krebs cycle and inhibits the Krebs cycle. We don't want these two things to be happening in a fasting state. Why? Because we don't have enough glucose to do it. All right, so what do we want to do then in this fasting state? We want to increase our glucose levels. But if we don't have any, what can we do? Well, the very first thing we do is break that glycogen down. Now, glucagon is a stimulator of this process. Now, a great thing for us is that going from glycogen to glucose 1-phosphate, that's a reversible step. So the enzyme used here can be used to go backwards. Cool. From glucose 1-phosphate to glucose, that's a reversible step. So that can go backwards. But the problem is going from glucose 6-phosphate to glucose, in order for that glucose to be able to, in this case, leave the bloodstream to increase it, it can't. That's irreversible. But here's the great thing. Glucagon, when it goes up, stimulates the transcription and translation of enzymes here that allow for this reverse to happen. So glucagon allows for an enzyme here to be produced, and this enzyme is called glucose 6-phosphatase. Glucose 6-phosphatase, and I'll write it like that, just because it's a big term. That is mediated by high levels of glucagon. So that means now we can take glycogen, make glucose, throw it into the bloodstream. That's exactly what we do 3 to 4 hours after eating. We start to break it down slowly. But the thing is, it doesn't just use glycogen. Simultaneously, it will use that predominantly in the front end. So between 3 to 8 hours, it's predominantly of fasting. It's predominantly using glycogen. But it's also starting to utilize non-carbohydrate-based sources to produce glucose, to try and increase that blood glucose level. Non-carbohydrate. That means it's using sources of energy that don't fit into this pathway. They have to be fed into that pathway. So one, for example, that we need to talk about is lactate. This is probably the most utilized non-carbohydrate-based source for energy in a process called gluconeogenesis. If you think about the term gluconeogenesis, gluco means glucose, neo means new, genesis means the beginning of. Think about that backwards, the beginning of new glucose. That's what we're trying to make here from non-carbohydrate based sources. First one I want to talk about is lactate. Now, what is lactate? Where does it come from? Sometimes people term it lactic acid, which I think is a misnomer. Lactate is more accurate. Lactate can come from muscle, so remember, muscle needs glucose. uses glucose for energy, and to undergo this whole process. But one thing I didn't tell you here was that we might use all these electrons and protons to generate ATP, but then what happens to all the electrons and protons that are left over? Electrons are damaging to the body, right? Protons make the area or the environment acidic. We've got to get rid of this stuff. What do? We have oxygen. The great thing is we can take oxygen, and oxygen will bind to the electrons and protons. And what it's going to do is it's going to produce, if we snap hydrogen and oxygen together, what do you think it produces? Produces water. Innocuous water that our body needs, but it requires oxygen. So this whole process will only happen all the way to the electron transport chain if oxygen is available. If oxygen isn't available, that backs up. This backs up, and it backs up to pyruvate. Now, this is the thing. In the muscle, when we are doing sprinting or we're lifting weights really fast, really quickly, in a way that we're trying to produce more, we need more ATP than we can make with the available oxygen, so we don't have enough oxygen, basically. This all backs up to pyruvate, and pyruvate turns into lactate. This is in the muscle, not in the hepatocyte here, but in the muscle. So muscle will produce lactate. Now, the great thing is that lactate will jump into the bloodstream, enter the liver cell, and will turn back into pyruvate. Brilliant, because in the liver, pyruvate can turn into glucose. So in the muscle, that lactate is useless. It can't turn into glucose for energy. So it must jump into the bloodstream. So from the muscle, the lactate jumps into the bloodstream, enters in the liver, and goes from lactate to pyruvate. Now the thing is, you might be looking here and going, yeah, but going from pyruvate to phosphatidylpyruvate, that's irreversible. We can't go from there to there and then go all the way back up, right? So what happens? All right, let's take a look. When muscle makes lactate and turns into pyruvate, pyruvate will enter the mitochondria and will turn into acetyl-CoA. No, it won't. Pyruvate will turn straight into oxaloacetate. Now, this is where that glucagon comes into play. Remember I told you it switched on glucose 6-phosphatase? Glucagon will switch on an enzyme here called pyruvate carboxylase. Pyruvate carboxylase, which turns pyruvate to oxaloacetate. Pyruvate to oxaloacetate. This uses ATP. We need ATP in this process. Now, oxaloacetate can turn into malate. That's no problem. And what happens is malate will leave. Go back into the cytosol. And what malate will do is malate will turn back into oxaloacetate. Now, you might think, wait a minute, oxaloacetate to malate, malate back to oxaloacetate. Why doesn't oxaloacetate just leave? It can't. Because of its structure, oxaloacetate cannot leave and pass through the membranes of the mitochondria. But remember here I said when malate goes to oxaloacetate, it produces NADH. But when oxaloacetate goes to malate, it goes backwards. So that also means when malate goes to oxaloacetate, NADH, so again, reversible. I'm not going to write it up. There's no point writing it up because it's happening there. But the thing is, oxaloacetate doesn't turn to pyruvate. It turns into phosphatidylpyruvate. So as you can see, when blood glucose levels are low and glucagon levels are high, glucagon stimulates glucose 6-phosphatase to allow for glycogen to be produced into glucose. It produces pyruvate carboxylase for that to happen and pushes it down into this process and stimulates this enzyme here, which is called phosphatidylpyruvate carboxykinase. That's a great name, isn't it? We can write phosphatidylpyruvate, PEP, carboxykinase. And so that turns to phosphatidylpyruvate, and then look, it can go backwards, 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 backwards. Uh-oh, can't go backwards here. It's okay, glucagon allows for another enzyme to be released so that it can go backwards here, and that's called fructose. I've wiped that out accidentally. Fructose 1,6-bisphosphatase. Fructose 1,6-bisphosphatase. Now, what can we do? We can take lactate and turn it to pyruvate. Pyruvate can turn into oxaloacetate, which turns to malate, which turns back to oxaloacetate, which turns to phosphenylpyruvate, which goes back, glucose. Lactate can turn into glucose, and that can jump into the bloodstream and feed back to the muscle cell itself. Awesome. Now, muscle isn't the only producer of lactate. Red blood cells produce lactate as well. So they both produce lactate. Now, why red blood cells? Remember, red blood cells they're just packed with oxygen. They have no mitochondria. If they've got no mitochondria, this doesn't happen. Ironically, red blood cells might be packed with oxygen, but it can't use it for the electron transport chain because it doesn't have one. So all red blood cells can do is glycolysis. What's the end stage of glycolysis? Pyruvate. What are you going to do with pyruvate? When you're done with it, turns to lactate. But remember, red blood cells can't. So when glucagon, making these really important enzymes here, this only happens in the liver and the kidneys. It doesn't happen in the muscle, it doesn't happen in the brain, which means this process of gluconeogenesis, which ultimately is taking pyruvate to make glucose again, it only happens in the liver and kidneys. That's why when muscle and red blood cells make something like lactate, it has to deliver it to the liver so the liver can undergo gluconeogenesis. Super important point. Super important. All right, so we've got lactate. That is an energy source in this process. You can also use amino acids as well. And amino acids come from proteins and muscle, and one important amino acid is alanine. It's probably the most utilized amino acid for gluconeogenesis, and it again jumps to pyruvate, which we've spoken about, that process. But there's other amino acids that can jump in, like glutamate, for example. We know that glutamate can be made from a range of other amino acids too, but that can turn into alpha-ketoglutarate. And there's amino acids that can jump in here and amino acids that can jump in here, for example, and other discrete areas. So amino acids can be used as a substrate to make glucose, to increase blood glucose levels. Here's another point. We've also got triglycerides, right? Triglycerides, or triacylglycerides, depending on what you want to call them. That's fat, right? It's in adipose tissue predominantly. And triglycerides are made up of glycerol and fatty acids. So the triglycerides will split off and release the glycerol and will release the fatty acids. All right, let's first look at glycerol. Glycerol, thankfully, can turn into dihydroxyacetone phosphate. Now, what does that mean? Glycerol, dihydroxyacetone phosphate can go back. Yep. Glucose. Glycerol can turn into glucose. So not only is lactate a source and amino acids a source, but glycerol is a source for gluconeogenesis. These are the three major gluconeogenic sources: glycerol, lactate, and amino acids. You might be thinking, what about fatty acids? I'll get there in a sec. But let's talk about fasting, right? Let's say you've woken up from sleep. You've slept 8 hours, you haven't eaten in 8 hours. So you've been fasting for 8 hours. After this time, what you're going to find is that after this 8 hours, glycogen will be contributing around about, let's use blue, glycogen will be contributing around about 40 to 70% of the glucose production after 8 hours. Lactate will be contributing around about 7 to 18%. Amino acids like alanine will be contributing around about 5 to 11%, and glycerol will be contributing around, other way around. Glycerol will be contributing around about 5 to 11%, and amino acids will be contributing around about 3 to 5%. So as you can see, outside of glycogen, lactate is the major gluconeogenic substrate. That's important. Now that's after 8 hours. What do you think happens after 14 hours, for example. So after 14 hours, what you're going to find is that these gluconeogenic substrates, they will be contributing 54% of the glucose. After 22 hours of fasting, they move up to contributing to 64% of the glucose, which means glycogen's dropping, right? After 42 hours, so we're nearly at 2 days here, gluconeogenic substrates contribute 84% of the glucose. Because again, the point is to increase blood glucose. Why? Why do we want to increase blood glucose? Why don't we just want to make energy? Because blood glucose levels are important because that's what the brain uses. That's what red blood cells use. That's predominantly what muscles will use. That's also what the retina and renal medulla uses. So we need the glucose levels to be normal in the bloodstream. That's important. Because if you think about this process, taking pyruvate, right, because ultimately a lot of gluconeogenesis is going from pyruvate through, this uses energy. You can see energy is used here, for example. Energy is going to be used here. We take GTP and turn it into GDP. So we use energy, we use energy. We also use energy here, right? By reversing this, we take ATP and turn it into ADP. Here we take NADH and turn it into NAD+. So as you can see, we lose ATP here, we lose ATP effectively here, we lose ATP here, we lose NADH here. So when we look at the process of gluconeogenesis, net, you end up getting, you lose 1, you lose 2 ATP, you lose 3 ATP, and you've lost an NADH. But remember, we've got 2 of these. So we lose a lot, right? Let's just say if we do it from 1, we end up losing 2 ATP. We lose around about -2 ATP, -1 GTP, and -1 NADH. But my point is that gluconeogenesis needs energy. It needs energy. And you might be thinking, but wait a minute. My blood glucose levels are low. I've got no energy. I'm fasting, right? I'm 14, 22, 42 hours of fasting. I need energy. I don't have energy. So how can I undergo gluconeogenesis to make glucose if I don't have any available energy? Well, this is where the fatty acids come into play. The fatty acids will turn into acetyl-CoA. At least the even chain fatty acids will. This process is called beta oxidation. And the great thing about beta oxidation is it produces NADH and FADH2, which I told you, what do? They produce ATP. So effectively, you can, and this changes depending on the fatty acid, get around about 4 ATP molecules just in the process of beta oxidation, taking fatty acids to turn into acetyl-CoA. This is the energy that's used to feed gluconeogenesis. Hopefully that makes sense. But let's think even more long term, right? We're going 48 hours and above. Think about this. If we want to make glucose, and ultimately we're doing it by turning pyruvate into oxaloacetate, but oxaloacetate is getting dragged out of here, right, in the form of malate. So to create glucose, think about the Krebs cycle. We're pulling oxaloacetate out. But I told you we need oxaloacetate to bind to acetyl-CoA. When we break down fats, which we do when we're fasting, fatty acids are turning into acetyl-CoA. So we're increasing our acetyl-CoA levels, but decreasing our oxaloacetate levels because it's being taken out and turning into glucose. Acetyl-CoA needs to bind to oxaloacetate to make energy, but it can't. So acetyl-CoA just starts to accumulate. All these fatty acids turn into acetyl-CoA, accumulate, and they snap together. Now, when acetyl-CoA snaps together, it forms something called ketones. like beta hydroxybutyrate, for example. That's a ketone. What the ketones do is they can jump into the bloodstream and go to the brain. The brain will take those ketones. Now, let's pretend that this is the brain. It takes those ketones, throws it into the mitochondria and turns it into acetyl-CoA. So it can enter the Krebs. But you might be thinking, wait, the oxaloacetate's gone. Not in the brain. Remember, only in the liver does this process of gluconeogenesis occur. So it's only in the liver where the oxaloacetate levels are low because it's trying to make glucose. In the brain, you're going to have normal oxaloacetate levels. But the problem is you're going to have low acetyl-CoA levels in the brain because you don't have any glucose to make it, right? So luckily, ketones can make acetyl-CoA, and then you can make all the energy that your brain, well, not all of it, because ketones are a bit of a sluggish way to produce ATP, but it can make ATP. So that is the difference between fed and fasting state metabolism. I'm Dr. Mike. I hope that made sense.
Hi everyone, Dr. Mike here. If you enjoyed this video, please hit like and subscribe. We've got hundreds of others just like this. If you want to contact us, please do so on social media. We are on Instagram, Twitter, and TikTok at Dr. Mike Todorovic, at DrMiketodorovic. Speak to you soon.